Hello everyone, my name is Sonali. Welcome to our channel Success EduTech. In the previous video, we had discussed about a natural method of vegetative propagation. To see that video, please click on I button or link is given in the description. In this video, we will discuss about artificial method of vegetative propagation. See, what is artificial method? There are few methods which are not occur naturally in the plants. Agriculturalists and horticulturalists have developed several techniques for the propagation of desired varieties of economically important plants and to multiply fresh stock of the plants. Means for the garden cultivation and the management, we require a plant with desired varieties like disease-free plant, healthy plant, and we have to multiply fresh stock of the plant for the propagation of that species. As per the human requirement, various methods are implemented. They are as follows: cutting, grafting, budding, tissue culture. Now, we will see first cutting. Are the small pieces of the plant parts like root, stem or leaf which are able to develop into new plants when placed in the moist soil. Means we have to cut small pieces of the plant parts like root, stem, leaves, and when we place into the moist soil, it will develop into new plant which is genetically identical to their parent plant. The cutting must have a primary meristem. Now, what is meristem? Meristem, as we know that meristem is a type of tissue occurs in the plant. It consists of undifferentiated cells capable of the cell division. Now, meristematic tissue is divided into apical, lateral and intercalary. Here, primary meristem means apical meristem are the meristematic tissue located at the apices of the plants. For example, root apices and shoot apices. Cutting is most convenient and cheap method of the propagation. Diagram of cutting. In the stem cutting, we have to cut 4 to 5 inches long stem in a slanting position for the successful rooting from the parent plant for the manipulation of the new plant. Don't break these top leaves because these leaves provide energy for the successful rooting. We have to transplant into the bed, or you have to transplant into the proper container for the nutrition. Don't transplant directly on the landscape. Here, after growth, then we have to transplant into the landscape. Now, in the root cutting, we have to cut the plant part from here and we have to transplant into the medium to provide nutrition and the growth for the root, and it will form a shoot here. Means the growth will develop here. Now, second method of the artificial method is the grafting. Grafting is a technique of joining together the parts of two different plants in such a way that they unite and continue their growth as a one plant. Means in this method, we have to take the two parts of different plants and we have to join, we have to unite them in such a way that they will grow in the form of one plant with the proper desired characteristic. In grafting, the plant rooted in the soil and on which the part of other plant is inserted is known as stalk. And while the other which is inserted on the stalk, the part which we inserted on the stalk is known as scion or the graft. This is very important point in the grafting. Now see the diagram of grafting. In the grafting, what we have to do, this is the root stalk cut for a grafting. We have to cut from here and we have to cut in the V-shaped. We know it is as V-graft. This is known as the stalk in which we have to insert the another part of the plant that is known as scion, and we have to wrap this portion, graft junction, wrap, and bud will shoot in the spring. The result will be shown in the spring. Now, budding is one kind of the grafting in which a single bud with a small part of the bark and living tissue is grafted on a particular stalk, or we can say in the budding, leaf bud is taken from one plant and grown onto the another plant. In the grafting, the cambium is very important plays important role. The cambia of both stalk and scion fuse together and make the union of these two plants successful. Now, what is this cambium? Cambium is a cellular tissue from which phloem and xylem or cork grows division result in the secondary thickening. Means the cambium of the stalk also and cambium of the scion also fuse together and they will form a new plant which will successful plant. Grafting is not possible in monocots because the cambium is not present in the monocots like dicot, means the monocots lack the cambium tissue. See the diagram of the budding. See, this is the leaf bud. Leaf bud. We have to cut this part and in such a way that we have to insert into the stalk. This is the cutting part of the stalk. We have to insert this leaf bud into the stalk. This is our scion and this is our stalk. And after inserted into the stalk, bud has developed here. The advantages of budding: the species that can't propagate by the cutting, we have to propagate that species by using grafting and the budding method. Now, last method is tissue culture and the micropropagation. Tissue culture is very important method for the propagation of the species of the plants. It is a method by which a small amount of plant tissue is carefully grown to give a many plantlets. We have to take very care while doing this tissue culture method. Micropropagation method is also used nowadays. For this method, we require a shoot apical meristem as an explant or mother plant, and many shoot apices can be produced which are called micropropagules. These micropropagules are used to produce large number of genetically identical plants, that is clones, within a short period of time. Means in a less time we have a great advantages by using a tissue culture method. See the diagram of tissue culture. Here the potted plantlet is here and we require in tissue culture a shoot apex, a tissue from the shoot apex. We have to take the shoot tips or meristem. Here the tissue are able to divide into the another layer. We have to keep into the primary culture. Now, what is this primary culture? Primary culture is made up of the agar gel, which is provide proper nutrients to this tissue for their growth and enzymatic maintenance also. Then that tissue will proliferate, means increase in the number and will develop into the shoot development. Then after shoot development, rooting system will be start, then rooting of the shoots. Then again this rooted plantlet will develop here. Again this plant we have to pot it into the potted and this will be the potted plant develop here. So this is very important technique for the micropropagation. Now see the significance of the vegetative propagation. Vegetative propagation is very significant as it is easy and cheaper method of multiplication. It is result in the genetically formation similar plant, thus improved variety can be multiplied. The yield can be increased by using all above method. Means by using all above method, cutting, grafting, tissue culture, we can increase our species propagation yield in a very short period. By this method, rapid propagation of plant can be done with the seed with a long dormancy period. Means the growth period of the seed will be long and healthy, so we can do the rapid propagation of plant. This method helps in the production of the clones of the economically useful and rare plant. Means we can develop the plants which are very economically important, and the rare plants also can produce by using all above method and the tissue culture method also. If you like my video, please share, like and subscribe to my channel. Thank you very much.